The first effector function is neutralization of virus or toxin. Antibody can bind directly to soluble toxin or virus so that the virus or toxin cannot reach to the host cell. So prevent pathogen host binding. So first is neutralization. Second thing is opsonization. In this case, a pathogen is coated with antibody and macrophage or any phagocyte that express Fc receptor against the antibody Fc region can engulf the bacteria very efficiently. Ultimate result is phagocytosis, but the process is called opsonization. So there is a difference between when the bacteria is coated with antibody or not, because when it is not coated, the efficiency of phagocytosis is low, but when it is coated, the efficiency of phagocytosis increases. So there are some molecules that are responsible for opsonization. They are called opsonin. The C1q protein also a opsonin, MBL managed binding lectin a opsonin, phycolin a opsonin, and also antibody can perform opsonization process. The next is complement activation. When a tumor cell or virus infected cell is coated with antibody, what happens? Antibody can recognize any viral infected cells or tumor cells because the tumor cells express some new antigen. The cell is coated with antibody and C1q protein can bind with that antibody, like it bind with IgM and IgG molecule antibody. It starts a process called complement pathway, and the ultimate result is the formation of MAC molecule, membrane attack complex, that actually form a pore-like structure and rupture the cell membrane, and the ultimate result is lysis of the cell and the cell die. The fourth major effector function is ADCC, antibody dependent. So it is depend on the antibody. This whole process is depend on the antibody. Cell mediated, because NK cell take part into the cell mediated immunity, and cytotoxicity means the NK cell actually kill the target cell directly. So NK cell is actually cytotoxic in nature. They can secrete perforin and granzyme and kill the cell directly. So these four, like neutralization, opsonization, activation of complement pathway, and ADCC, are the major four functions of antibody you must remember. So this is the opsonins, example like C1q, MBL and phycolin. They bind microbial surface molecule. C1q protein can directly bind with this microbial surface antigen, or any antibody that is already bounded with the antigen. The C1q protein can bind with two type of antibody like IgM and IgG. MBL protein, managed binding lectin, can bind with mannose or fucose with high affinity, and the phycolin can bind with any acetyl oligosaccharide, acetylated oligosaccharide like N-acetyl glucosamine. And you can see the similarity between these three structure. They looks very similar, perform similar work, but bind different molecule. MBL bind with high avidity. Why it is high avidity? Because it is trimeric, so it's actually polyvalent in nature. 15 binding sites are already there, so it is polyvalent interaction, so it is high in avidity to manage and fucose residues. The phycolin can bind with any oligosaccharides containing acetylated sugar. Now we can see the same functions of antibody but with a different diagram. So these are all antibody responsible for particular function. Let's assume that the yellow antibody are responsible for the neutralization. So if there is no antibody, the bacterial toxin can bind with the receptor and activate the cell or perform the toxicity. But if there is antibody specific to that toxin, it can bind with the antigen, or it can be virus, so that the toxin or the virus cannot reach to the host cell. So this is neutralization. Another situation is called opsonization. In this case, the bacteria is in the extracellular space. If the bacteria is in extracellular space, then opsonization can happen. The antibody that can recognize the bacteria or antigenic determinant or epitope can bind, and the bacteria is coated with antibody. Macrophage express some receptor that bind with the Fc receptor. But if the bacteria invade the plasma or our bloodstream, then in the blood plasma some molecules are present, soluble molecule like complement protein and also antibody. So the antibody coat the bacteria, or the complement protein directly start complement activation. After neutralization, the macrophage can engulf or ingest the toxin or bacteria. In this case also, the macrophage ingest the bacteria with high efficiency. That's opsonization. Greek meaning is to make tasty. And complement pathway, and the result of this complement activation is formation of a pole-like structure called MAC, membrane attack complex, that form a pole-like structure, and the ultimate result is lysis. When a cell is infected by a virus or a tumor cell, antibody can bind with antigen, and the NK cell express a special type of receptor that can bind with the Fc region of antibody. Especially the NK cell express receptor for IgG1 and IgG3. After binding with this antibody, NK cell sense that these cells have some problem and it can directly kill the cell.